Hello everyone, I am Dr. Tulsidas Patil, currently working as Assistant Professor in Department of Pharmaceutics at Sri Vile Parle Kelvani Mandal's Institute of Pharmacy, Dhule. In this video, I am going to discuss about additives utilized in parenteral products and the importance of isotonicity. Disclaimer: this presentation is made for education purpose only and not meant for any commercial interest. The information which is provided in the presentation is taken from the own experiences of the presenter and his understanding and from the references that are quoted at the end of this presentation. Learning outcomes: after successful completion of this topic, students will be able to understand about the additives utilized in parenteral preparations and the importance of isotonicity. So let's start with the topic. First we will see the purpose behind incorporation of the additives in the parenteral formulations. Additives, which are also referred as excipients, can be added in the parenteral formulations to maintain pharmaceutical stability, to control the product attributes, to ensure the sterility, and to aid in the parenteral administration. There are different categories of excipients or the additives that can be added within the parenteral formulations. These are solubilizing agents and surfactants, antioxidants, antimicrobial agents, buffers, chelating agents, suspending agents, protein stabilizers, bulking agents, protectants, and tonicity adjusting agents. So let's see the information about different categories of excipients and their suitable examples. First one is the solubilizing agent and surfactants. We can increase the solubility of our drug by utilizing various solubilizing agents. The examples are like ethyl alcohol, glycerin, polyethylene glycol, propylene glycol in the concentration range of 1 to 50%. We can also utilize surfactants in dispersion systems. They have different roles, like they used to disperse the water insoluble drug as a colloidal dispersions. They are utilized for wetting and preventing the crystal growth in the suspensions. They are also utilized for acceptable syringeability. Examples are polyoxyethylene, utilized in concentration range of 0.1 to 0.5%, and sorbitan monooleate in the concentration range of 0.05 to 0.25%. We can utilize lecithin as a emulsifying agent in the emulsions for solubilizing the steroids and fat soluble vitamins. Other examples of solubilizing agents are polyoxyethylene castor oil derivatives and cyclodextrins. Polyoxyethylene castor oil derivatives are utilized to solubilize the hydrophobic drug, and this can be utilized in formulations of paclitaxel, diazepam, and cisplatin. Cyclodextrins are nothing but the cyclic oligosaccharide molecules which are having bucket-like structure in which they contain hydrophobic central cavity while outer surface is hydrophilic in nature. These cyclodextrin molecules form the inclusion complexes with variety of poorly water soluble molecules. We can utilize alpha and gamma cyclodextrins in parenteral products. However, beta cyclodextrins cannot be utilized because they may cause severe kidney damage. The next category of excipients is antioxidant, which are added to prevent the drug from oxidative decomposition. The examples of antioxidants are ascorbic acid, cysteine, monothioglyscerol, then salts of sulfur dioxide, BHA, BHT, and tocopherols. We will see some brief information about these antioxidants. Ascorbic acid, we can utilize it in aqueous parenteral products in the concentration range of 0.01 to 0.05% weight by volume, and we can also utilize it for adjusting the pH of the formulation. We can utilize alpha tocopherol in oil-based parenteral products in the concentration range of 0.05 to 0.5% volume by volume, as it is highly lipophilic in nature. Other examples of antioxidants are BHA and BHT, which are utilized for intramuscular injections in 0.03% weight by volume concentration and for intravenous injections in 0.000 to 0.002% weight by volume concentration. Then salts of sulfur dioxide, which includes bisulfites, metabisulfites and sulfites, can be commonly used as antioxidants in aqueous parenterals. The examples are sodium metabisulfite in 0.1 to 1% concentration for acidic parenteral products. We can use sodium bisulfite for the products of neutral pH, whereas sodium sulfite can be utilized as an antioxidant in alkali parenterals. However, sulfites may cause allergic reactions in some asthmatics. The another class of additives is antimicrobial agents. Antimicrobial agents are utilized in all multiple dose parenteral products for inhibiting the growth of microorganisms accidentally introduced during the withdrawal of individual doses. However, they should not be added to large volume parenterals, those are infusions, or the products which are intended for intraspinal or intraocular injections. Examples of antimicrobial agents are benzalkonium chloride, benzyl alcohol, chlorobutanol, metacresol, then butyl parahydroxybenzoate, methyl parahydroxybenzoate, propyl parahydroxybenzoate, phenols and thiomersal. Preservatives may be added to single dose parenteral products also, which are not terminally sterilized. However, interactions in between these preservatives and other components of the formulation we need to consider, because that may compromise the efficacy of the preservative. The next category of additives is the pH adjusting agents and the buffers. So injectable products should have a pH value in between 3 and 9 prior to the administration. pH above or below this range are too corrosive and may cause tissue damage at the site of injection. pH can be adjusted by utilizing acidifying agents like hydrochloric, citric acid and sulfuric acids, and alkalizing agents like sodium bicarbonate, sodium citrate and sodium hydroxide. The changes in pH may arise because of the interactions in between ingredients in the formulation and the container, or from changes in storage temperatures. So buffers are another category of excipients. Those are utilized to maintain the pH of the product at desired optimal value. The examples of buffers are like acetate buffers, citrate buffers, phosphate buffers, with typical names are like citric acid, sodium citrate, sodium acetate, sodium lactate and mono and dibasic sodium phosphates. These are utilized in concentration range of 0.8 to 2%. Amino acids we can also utilize as a buffers, especially for polypeptide injectables. The next category of additives are chelating agents. These agents will form the complex and thus inactivate metals such as copper, iron and zinc, which are generally catalyze oxidative degradation of the drug molecules. Example that is most commonly used is salt of ethylene diamine tetraacetic acid, that is EDTA, in 0.01 to 0.05% and citric acid or tartaric acid. Another additives are suspending agents. These are the additives which will impart viscosity to the product and slower down the settling in suspension type of parenterals. Examples of suspending agents that can be utilized are methyl cellulose, carboxymethyl cellulose and polyvinyl pyrrolidone. Then protein stabilizers are the additives. Those can be utilized to stabilize the proteins and peptide drugs against their aggregation and degradation. The best examples are amino acids such as glycine, lysine, etc. Then bulking agents are the category of excipients. Those will increase the solid content of the reconstitutable freeze-dried products. These are water soluble in nature and provide porous solid mass. The best examples are mannitol, lactose, sucrose, trehalose, etc. The next category of additives are protectants. These are the substances those will protect the formulation against the loss of its integrity or reactivity. Examples are cryoprotectants, lyoprotectants, and those which will protect the formulation against the adsorption. First, we will see the cryoprotectants and lyoprotectants. These are the excipients which will be utilized to inhibit the loss of integrity of the active substance, which will be resulting from freezing step and freeze drying step respectively. Polyethylene glycol is the example which will protect the lactate dehydrogenase and phosphofructokinase from damage by freezing step, but they cannot protect either of the proteins from damage by freeze drying. For this, we have to utilize sucrose and trehalose for both the proteins. Then protectants can be utilized against the adsorption of the material or the equipments in the manufacturing process. Human serum albumin is example which can be utilized as a protectant against the adsorptive loss of proteins. For example, more specifically, we can utilize 0.1 to 1% human serum albumin to prevent the adsorptive loss of insulin to hydrophobic material. Now we will see the importance of tonicity and the use of tonicity adjusting agents. First, maintaining the isotonicity of the product is utmost important to avoid any swelling and contraction of the blood cells. Aqueous solution of sodium chloride at concentration of 0.9% weight by volume or 9 gram per liter has osmolarity of 286 millimole per liter, and which has osmolality in between 280 to 295 millimoles per kg, is considered as isotonic, which means has the same osmotic pressure with the human plasma. Hypotonic solutions have lower osmotic pressure than the plasma. If we mix with them with the blood, they will cause the blood cells to swell and burst, as water will be driven into the cells by the process of osmosis. Hypertonic solutions have higher osmotic pressure than the plasma. So if we mix them with the blood, they will cause the blood cells to lose the water by osmosis and shrink. So, hypotonic injection solutions are made isotonic by addition of sodium chloride, dextrose or potassium chloride. The hypertonic injection solution must be made isotonic by dilution prior to the administration. So there are different methods which can be utilized for adjusting the tonicity. Few are like freezing point depression and use of sodium chloride equivalents. These are the key references which are utilized for making this video presentation. Thank you.